Hello and welcome to my Chapter 3 supplemental lecture on the plasma membrane and the moving of materials through that membrane. So the plasma membrane in cells is made up of what's known commonly as the phospholipid bilayer. And if you recall from Chapter 2, phospholipids are a type of fat. And these fats are very specific in their structure in that they have a head that's polar, so that polar head interacts with water, and they have two fatty acid tails that are hydrophobic and do not interact with water. So this very specific structure of these phospholipids means that they interact with water in a very specific way, in that if I have water sort of at the top here, and I have my phospholipid head with the two fatty acid tails, the polar head interacts with water where the tails are repelled by the water. Conversely, if I have a layer of water and say air here, the polar head represented by my hand and the phospholipid tails will arrange in this sort of fashion automatically, simply because of just the interactions between the hydrophobic and the hydrophilic portions of these phospholipid molecules. That very specific arrangement is the reason why cell membranes self-arrange in the way that they do. It's a very specific interaction between these molecules and water. These molecules will arrange, if this is one molecule, another molecule will arrange itself like that, and then the next one here and here, all the way across, and they'll form these sheets of phospholipid. So if you were to pour liquid phospholipid on the top surface of a water, you'd end up with a series of these phospholipids with their polar or their nonpolar tails sticking up out of the water in this self-arranged fashion. Now, if you put enough of these phospholipids in place in the water and then maybe surround it by water, they will form a sphere, because the polar heads will interact with the water and the hydrophobic tails will repel away from the water and they'll form this spherical shape. If you then add some more and a little more structure, you can build what's called this phospholipid bilayer, where you have the fatty acid tails, hydrophobic, will come together like this with their polar heads with water on either side. So water in the cytoplasm, water in the environment. This hydrophobic layer on the inside of the plasma membrane is built with these fatty acid tails. What this means is that this self-arranging structure of the cell membrane will build itself it will do so spontaneously. It doesn't require any additional energy by the cell to maintain the structure of the cell membrane, and this is a key component to the building of cell membranes. Also incorporated within cell membranes are additional structural proteins and enzymes and carrier proteins and channels and glycoproteins, glycolipids, other associated sorts of chemical structures. And these all interact together and build what's known as the fluid mosaic model. Because these phospholipid heads are not, they're in contact with one another, but they're not necessarily attached. They're simply being squeezed together by water. And so that alone is not a very stable connection. And so the addition of these proteins maybe cause these linkages so they move less. But the cell membrane is a very, very dynamic system, and it's important in understanding that this fluid model for the cell membrane is really what allows a lot of the flexibility within the cell to occur. I'm going to try and give you a good visualization of sort of the general structure of this phospholipid bilayer, and I'm going to use glass marbles as my model. So imagine that these glass marbles are my phospholipids. I have one, and they will sit side by side next to one another, and they will self-arrange in these sheet sorts of patterns. If we look at this small tray of marbles as representing a flat sheet of phospholipids, we can see that these phospholipids can move freely in space around one another, even though they're still touching each other. So this is, when we're talking about the fluid model, this is what it means. It means that these things can move along and beside each other freely. They're not physically attached. But again, it's not that there's just phospholipids in the phospholipid bilayer. There are also integral proteins, fats, glycoproteins, carbohydrates, any manner of these other sorts of ancillary structures within this phospholipid bilayer that adds to the stability of these phospholipids. It's important to note that represented by a single tray of marbles here, this is a single layer, so this would be like a phospholipid monolayer, not the bilayer that we see with cell membranes. But what we can do is that if we simply add some marbles on top, oops, I've lost my marbles. Ha. If we add marbles on top, we can see that we start to develop this more three-dimensional structure within the broader structure that we can see. And so as we add more and more marbles to our layer, we can see that we've developed this section here that's 2 marbles sort of thick here and here, along with these integral proteins and other associated structures with this phospholipid bilayer. So I'm going to try and move this around so you get a better view of it. But you can see that there's this definite three-dimensional sort of structure to the phospholipid bilayer. I hope that helps clarify a little bit about the physical structure of the plasma membrane, and that it builds this three-dimensional shape independent of any energy expenditure by the cell. This is something that spontaneously happens simply because of the interactions with water and these hydrophobic elements of the phospholipids. But because these cell membranes have this hydrophobic center to them, they do need to be able to move substances back and forth across the cell membrane. And this happens through a number of different mechanisms. Primary amongst these is the process of diffusion. And you guys are all familiar with diffusion. If you spray a spray of perfume in the air, there's a very concentrated area where the smell is intense, and as you move away from that scent dissipates. Also over time that scent dissipates, and that's diffusion, as those molecules don't want to be close together and they spread out and they become evenly distributed in that space. That's basically diffusion. So moving from areas of high concentration to areas of low concentration. And that, again, requires no energy on the part of the cell. That's just the thermodynamics of these molecules moving in their environments. Diffusion is an important way that things move across cell membranes. If something is too big to move across a cell membrane, it's just physically too big to squeeze between those spaces between the phospholipids, it's required that an enzyme or carrier molecule is in place to help facilitate that movement across, and so we call that sort of transport facilitated transport. Now, with facilitated transport, it still requires no energy on the part of the cell. It's still moving from areas of high concentration to low concentration, but it's simply using a carrier protein that uses that concentration gradient to drive things across the cell membrane. A specific type of movement through the cell membrane is called osmosis, and that's specifically the diffusion of water across a cell membrane. That term specifically for osmosis means diffusion across a semi-permeable membrane. So that's a good one to keep in your brains as well. We can see diffusion of water again when we talk about tonicity, and we have hypertonic and isotonic and hypotonic plant cells. You can see that in figure 3.2.3 of the book, where you have water moving into cells and causing them to swell, and the cell membrane then pushes on the cell wall and adds turgor pressure to provide support for those green sorts of structures in a plant. Now, if we're trying to move substances across a cell membrane, but we are moving them against the concentration gradient, so from low concentration to high concentration, and keep in mind that that's opposite of diffusion, then we need to have additional energy expended by the cell, and we call this sorts of transport active transport. Active transport requires the use of adenosine triphosphate, or this high energy molecule that cells use for pretty much all their energy needs, but it requires energy from the cell to push these things back into higher concentration areas. Again, think of if you had sprayed that perfume in the air, what it would take to collect all those perfume molecules and push them all back into a very concentrated space once you release them into the environment. It takes a lot of extra work to do that. It's not impossible, but it just takes a lot of extra work. In some cases, this active transport is accomplished by an enzyme that simply uses ATP and opens and closes and moves whatever it's moving against that concentration gradient. That happens very commonly in cells. It's also possible that cells need to move bulk materials through the cell membrane, so a chunk of something into the cell or out of the cell. If we're moving things into the cell, we call that endocytosis or phagocytosis, which translates to cell eating. So we're moving a chunk of something into the cell. So the cell membrane, remember, is flexible, and so it'll indent, and then, I can't move my hands that way, but it'll indent and then surround whatever it is the cell's trying to bring in and bring it inside of this newly formed vacuole and then or vesicle rather, and then digest it inside the cell with a lysozyme. Conversely, if the cell is trying to move something out, so if it's say a hormone producing cell and it's trying to move out vesicles of hormones out into the environment, the same thing occurs. These vesicles are formed on the insides of the cells and move to the exterior cell membrane, and that interior contents are then released out into the environment. Those are active transport methods that are used by cells to bulk move material in and out. I hope this helped clarify the structure of cell membranes and some of those transport mechanisms that we see with cells. These are important concepts when we're learning about how cells function and how they work. So I hope this was helpful. If you have any questions, again, please use the discussion boards to ask questions. If you want to email me, that's fine as well. Thank you.